So budding can be described as a form of what they call asymmetric mitosis or cytokinesis. Instead of the cell splitting into two equal halves, it splits into one large part and a smaller part. So budding. Now the two yeast that we're going to look at today, they're listed in little microprint on your handout. The first one is called Saccharomyces, and Saccharomyces is commonly called brewer's or baking yeast. Saccharomyces cerevisiae is the yeast we use to make leavened bread. It's also the yeast that's used to carry out alcoholic fermentation of grape juice to make wine, or of germinated grain to make beer. Yeah. So very important here in California, the wine, beer, bread industry. Okay. And then a second yeast that you'll be looking at prepared slides is the opportunistic yeast pathogen Candida albicans. So Candida albicans in folks that are immunosuppressed or maybe have been on broad spectrum antibiotics and wiped out all their good competing bacteria, often folks can develop Candida albicans infections of the mouth, which is commonly called thrush. We can develop Candida albicans infections of the anal rectal area. And women often will develop Candida infections of the vaginal mucosa, often following broad spectrum antibiotic use. Okay, so you'll be looking at slides of Candida as well today. Now, the molds are described as multicellular and filamentous. Filamentous means they grow in these long hair-like chains. So multicellular filamentous fungi. Okay, and these filaments, we can really think of them as a chain of cells. Chains of cells. And these little filaments, they have a special name. Single one is called a hypha. That's singular. And plural is hyphae. Now, the way I showed it here, it looks pretty big. If you saw a mold, say, growing on your bread, these hyphae, these filaments, they'd appear like little delicate white threads, like that. And in the molds, these hyphae, they tend to grow in these disorganized mats. And this mat of hyphae, it has a fancy name. It's called a mycelium. Okay, so if you left a piece of bread out in your kitchen, and maybe after a few days you came and you saw like these white threads on the surface of your bread, that would be the mycelium, this mat of tangled hyphae. Okay. And that's the characteristics of molds, is they make these disorganized hyphae. They don't form specific structures. Now, in contrast, the fleshy fungi, they share these same traits. They're multicellular, they're filamentous, but their hyphae are very well organized into distinct structures. They have well organized hyphae. And a great example of fleshy fungi are mushrooms. And just so we can understand what we mean by well-organized hyphae, okay, let's say we're looking at a cross-section of mushrooms growing in the dirt. So this would be the dirt. Okay, and the stalk of the mushroom, often referred to as the stipe, it's a collection of parallel hyphae. Okay, so the stalk of the mushroom is these very well-organized parallel array of hyphae. So this is what we mean by, in the fleshy fungi, the hyphae are very well organized. Oops, I don't have some good mushrooms here. Okay, we'll search for some. Okay. All right, so we have the yeast, the molds, and the fleshy fungi. Very good. Okay. Now, looking at structure, you guys, and I'm just kind of working my way down here, we want to take a look at the hyphae. We want to take a look at the different types of hyphae based on structure and function. So the fungal hyphae. Okay, so there's two ways to describe describe the fungal hyphae that we're going to use today. One is based on structure, and one is based on function. Okay, so if we take a look at your handout there under number 2, it says that these filaments, these hyphae, they can have two basic structures. They can be cenocytic, and I always forget how to spell the cenocytic, or another term for cenocytic is aseptate. And the other one, the other type of hyphae based on structure, they're called septate. Okay, so what's this? What is this septate and aseptate? Okay. Well, just so we know, down here, a septum is a cross wall. Okay, septum is singular. Septa is plural. So many cross walls. So what we mean by cenocytic or aseptate hyphae is that if we look at one of those filaments, one of those hypha, we won't see distinct cross walls between individual cells. It might be just a little tiny little indentation here. Okay, but these aseptate or cenocytic hyphae, they lack cross walls. So it's almost as if the hyphae, in being like a chain of individual cells, it's almost like one huge long cell, multi-nucleated, lots and lots of nuclei, because each of these compartments, which actually is a separate cell, would have a nucleus. Okay? Now, why is this a big deal? Well, mycologists use the structure of the hyphae to help them identify unknown fungi. So these cenocytic hyphae, these are considered, how would we call it? I guess more primitive. Okay, and we're going to find these in the class of fungi called Zygomyces. So you think, okay, these are more primitive fungi. They haven't evolved the sophistication of some of the other fungi. Okay, all right. And the example of a fungus that you will see today that has the cenocytic or aseptate hyphae is the bread mold called Rhizopus. Okay, Rhizopus. Now, according to the mycologists, the more evolutionarily advanced, the more sophisticated fungi have cross walls in their hyphae. So they're called septate hyphae. Septate hyphae. Okay. And again, if we zoomed in on one of the hypha with our microscope, we would see the cross walls or the septa. And what I'm trying to do here, you guys, is show the cross wall isn't complete. There's a tiny pore. There's a pore so that the cytoplasmic contents can move from one compartment to the next. Okay? Right? But the cross walls, the septa, are, again, help in classification. And these guys are considered to be more evolutionarily advanced. Evolutionarily advanced. Okay. And we'll see the septate hyphae in the group Ascomyces and Basidiomyces. Okay, so that's the two types of hyphae based on structure. Now, the second way we can classify the septa is based on function. All right? And for this, what we're going to do is, well, first of all, we'll describe the two different types of hyphae, and then we'll draw a little cartoon. So we have vegetative hyphae, and we have aerial hyphae. So what we'll do here is we're going to draw a little cartoon of Rhizopus. Okay, so we'll use as our example Rhizopus, the bread mold. And we can pretend this is Rhizopus growing on a piece of bread. So we'll put the bread here, the nutrients, all right?